Hello everyone and welcome to this virtual lab tour. I'm Sam, I'm a cancer researcher at the University of Manchester and I work for Stephen Taylor's lab, which is based at the Oglesby Cancer Research Building just opposite the Christie Hospital. Now before I start, I just want to let you know that I'll be wearing a mask and gloves throughout this video because I really want to protect my co-workers that are working at the moment. So what does Stephen Taylor's lab do? So traditionally we are a lab that looks at how cells grow and divide. And what's really cool about cells is when they attempt to divide, they have certain mechanisms or what we call checkpoints that have to be ticked off in order for that cell to divide successfully. So you've got one cell, it's mulling around, it's attempting to divide, it's ticking all of those checklists off, and then eventually we get two identical daughter cells. They both have the same amount of DNA with very little, if any, abnormalities. If they do have any abnormalities, what's also really cool about cells is they're able to self-destruct, so it minimises the chance of having abnormal cells growing in the body. But obviously, sometimes that can go wrong, and that's what leads to cancer cells. But what's really interesting about cancer cells and why I'm really interested in cancer cells is how they bypass these checkpoints. So I suppose let's try and use an analogy. So I'm going to use a computer game analogy. Bear with me. So a normal cell will go through all of these different checkpoints. So let's say certain tasks in a game. And you have to complete those tasks in order to complete that level. And then once you complete that level, you then go on to the next level. And that's kind of what happens with cell division. Ultimately, when the game ends, that's when the cell divides. But with cancer cells, they've got the game cheats that you used to be able to get off the internet. I know. Cancer cells have those. So those little game cheats that allow you to, find a secret route to bypass a really tricky part of the game, or certain cheats that allow you to finish the game really quickly. That's what cancer cells do. And obviously that leads to the cells to become cancerous and grow really quickly. And actually they survive with a lot of abnormalities because they're not checking off those lists, which would cause them usually to die. So what we do in the lab is try to understand exactly how those cancer cells get away with it, and also why they don't self-destruct when they've got all these abnormalities. Now, our lab's got some quite cool chemotherapy drugs knocking about, so we try and use some of these drugs in order to exploit these abnormalities and hopefully kind of push the cancer cell over the edge so that it will self-destruct. So it's kind of like gaining a superpower in order to defeat the powerful baddie at the end of the game. I hope you liked my video game analogy. Being next to the Christie Hospital is really, really useful for our lab because it means that we can get cancer samples straight from a patient. And we can grow these cells in a flask, just like this one. And it allows us to trial out different chemotherapy drugs and to see what drug kills or doesn't kill a patient sample. And the kind of long-term hope is that eventually one day we can lead to personalised medicine for each patient. So our lab has actually created a ovarian cancer biobank. So we now specialise in ovarian cancer. So this biobank contains lots of different patient samples, some from the same patient but just over the course of their treatment, which is really, really useful to see how the cells have adapted or changed over time and potentially become resistant to certain treatments. So how do we grow these cells? Well, now I'm going to show you what we call the tissue culture room, which is what I'm in right now, hence the blue lab coat, indicating that I'm ready to do some tissue culture work. So let's show you around. So this is the tissue culture room where I spend most of my time on a daily basis. I'm just going to start showing you this contraption, which is the tissue culture hood. So this is where we prepare all of our cells for experiments. Can you hear the noise? So that noise allows everything inside there to be sterile, aseptic, and just a clean environment, so it protects both me and my cells from any harmful bugs or bacteria. Now I'm just going to quickly pan over to the incubators. So we've got quite a few in this lab, and this is where we house the cells. So let me just have a little look inside here now. Okay, just so you don't get the reflection. So these are all of our patient cell lines that we're growing at the moment. Each flask corresponds to a different patient, which is pretty cool. And they all live at 37 degrees because we're trying to mimic the human body. Let's take a little sneaky peek at some of these patient lines down a microscope, shall we? Okay, so I've got my ovarian cancer patient flask ready to go, as you can see on this microscope. So let's have a little zoom in and have a look at these cells. So, as you can see, these here are all of the cells that have come from a patient, okay? So they're stuck to the bottom of the flask. That's why they're not moving around either. So one thing that is really characteristic of this patient line is how small these cells are, but also how they clump together, which is very, very interesting. So let me just show you another patient line and just see if you can spot the difference. Wow, so they look completely different, don't they? The cells are a lot bigger. We don't see them clumping together. And we also see that they're a little bit more elongated or longer, which is very, very interesting. So remember, these are two ovarian cancer cells. So they're the same cancer type, yet they look so different. But what I'm really interested in is whether a novel chemotherapy drug affects them the same or differently. So one way we can do this is to use a special microscope, a little bit more fancier than this one. And it lives inside an incubator, so that way we can basically grow the cells and track it over a period of time. But first I need to put some of these cells into a 96 well plate. And that's why I need to head towards the tissue culture hood. So now let's get ready, as you're about to get a tissue culture view of how I prepare my cells for a microscope experiment. Cue time lapse. So that plate is now in the incubator and it will sit there overnight, and then tomorrow morning I'll add some drugs and then stick it on the microscope. But in true Blue Peter style fashion, I've got a plate already prepared for you. So let's stick it on the microscope, shall we? So this is the fancy special microscope that I was talking about earlier, and it's called an Incusight S3. And this microscope is going to take a photo of the cells in every well of my plate every hour for 4 days, and this will allow me to track cell growth and also see how the chemotherapy drug affects the different patient cell lines. So let's open it up and stick my plate on, shall we? Right, I've pressed go, so I'll come back in 4 days' time and have a look and see how my cells are doing. Fingers crossed. Okay, that's me finishing tissue culture now. So I'm going to move into the main lab, but first I need to do a lab coat change because I don't want to cross-contaminate. Voila, now let's head into the main lab. So this computer holds all of the Incusight data, including my experiment. So let's fast forward 4 days so we can have a little look at the data from my experiment. But bear with me, all data analysis now happens at home. So I'm just going to head home. Right, so I'm in working from home mode now. I've demasked, I've degloved, I've got my specs on. I've remotely accessed the work computer via my laptop here. So yeah, let's analyse my data. So here is patient 1 and 2 side by side, treated with the same drug over 4 days. The IncuSight allows you to look at each individual well and measures how many cells are in the well at each time point, which is represented here by these graphs. So, if the cells are dividing normally, you would expect to see an increase in cell number at each time point. And this is what we see in well 1. This well has no drug in it. Therefore, these cells can grow really nicely, and you can see that the cell number increases at each time point over 4 days. From wells 2 to 6, this is where I added the drug, starting off with a very small concentration of the drug all the way to a high concentration of the drug. Let's look at patient 1 first. We can see that the growth is being affected at the drug concentration used in well 3, and it gets progressively worse as I add more of the drug. This would indicate that the patient line is sensitive to this drug. However, when we look at patient 2, the graphs seem very similar across all drug concentrations, meaning the cells seem to continue to grow regardless of the drug being present or when I increase the drug concentration. This patient line seems resistant to this drug. Now, another cool thing that the IncuSight can do is stitch together all of the images for each well into a movie. So now let's focus on patient 1 and see how 1, the cells grow nicely without the drug, and 2, what happens to the cells when we add a high concentration of the drug. The cells are labelled with green fluorescence, allowing the IncuSight to measure the cell growth accurately. Let's look at how the cells grow without any drug first. As you can see, the cells are dividing, they're moving around a lot, and by the end of the movie, they've divided so much that they fill up the whole well. This is a perfect example of growing cancer cells. Now let's look at the drug-treated cells. At first, they seem to move around a lot with some divisions. However, over time, we now see them slow down. Some start to get bigger, and some explode, indicating cell death. By the end of the movie, you can see that there are similar or maybe even less cells left in the well than there were at the start of the movie. This is very exciting to see, and I will analyse these cells further by performing different types of experiments to try and figure out what makes this patient line so sensitive. I reckon it's something to do with those abnormalities I was talking about before. Now, back to the lab. And that's Stephen Taylor's lab in a nutshell. I really hope you enjoyed the virtual lab tour. A big shout out to CRUK for funding all of the research that you've heard about today. And also my lab mates for helping me sort all of this out. Thank you. But for now, take care, stay safe, and I really hope that one day that we can do this in person. Bye.